Hi, my name is Päivi Saavalainen and I'm CEO of Cellex. Cellex comes from single cell X because we sequence single cells in XY coordinates directly from tissues or slides. And this is cancer. This is metastatic melanoma with immune cells and cancer cells. And for example, in cancer cell, the cancer research, the single cell resolution is really important. Because the cancer cells mutate all the time, so we need to study all of them in one by one in single cell resolution. And there are millions of them in the tumor, so we need a really high throughput to study all of them. And we need also spatial location of the cells to understand what they are doing in the tissue and what other cell types there are in the tumor, like immune cells, and what are they doing. And do they do their job? And the resolution with the current technologies is not single cell resolution. You can see that this is from the competitors' method. There are even tens of cells in each spot. In our method in Cellex, we have a much smaller spot size and much more of the spots, so we can really reach the single cell resolution. And we have an innovative way how to barcode the spatial information to these spots. We have a tissue slide with 160,000 of these tiny wells, and our patented barcoding method is based on color beads, which we can just see in the normal microscope scan. We have 20 different colored or sized beads tagged with synthetic oligotags, and once we spread these randomly to these wells of the chip, they can form up to 1 million different combinations, which we can record, and then later the RNA coming from the tissue and cells is linked also to this barcode information with these beads. And this method helps us really to see the single cell resolution of the cells in the tissue, and the researchers then understand much better how the cancer cells behave. And this can be also used in many other diseases and the basic research of different healthy tissues and diseases. This brings the real high throughput and quick analysis of hundreds of thousands of cells even. And it gives the location of the data and the whole transcript, all the genes which are expressed in different spots and areas of the tissue can be analyzed. So the data amount is huge to analyze. This data, the sequencing data of these hundreds of thousands of spots and for tens of thousands of genes is very deep and challenging, and so we need both AI models to analyze that and complex calculations, and we need data storage and calculation analysis capacity to analyze it.